Most E. coli strains are harmless, but some serotypes, EPEC, ETEC, etc., can cause serious food poisoning in their hosts, and are occasionally responsible for food contamination incidents that prompt product recalls. The harmless strains are part of the normal microbiota of the gut, and can benefit their hosts by producing vitamin K2, which helps blood to clot, and preventing colonization of the intestine with pathogenic bacteria, having a mutualistic relationship. E. coli is expelled into the environment within fecal matter. The bacterium grows massively in fresh fecal matter under aerobic conditions for 3 days, but its numbers decline slowly afterwards. E. coli and other facultative anaerobes constitute about 0.1% of gut microbiota, and fecal-oral transmission is the major route through which pathogenic strains of the bacterium cause disease. Cells are able to survive outside the body for a limited amount of time, which makes them potential indicator organisms to test environmental samples for fecal contamination. A growing body of research, though, has examined environmentally persistent E. coli which can survive for many days and grow outside a host. The bacterium can be grown and cultured easily and inexpensively in a laboratory setting, and has been intensively investigated for over 60 years. E. coli is a chemoatotroph whose chemically defined medium must include a source of carbon and energy. E. coli is the most widely studied prokaryotic model organism, and an important species in the fields of biotechnology and microbiology, where it has served as the host organism for the majority of work with recombinant DNA. Under favorable conditions, it takes as little as 20 minutes to reproduce. Biology and biochemistry. Type and morphology. E. coli is a gram-negative, facultative anaerobe that makes ATP by aerobic respiration if oxygen is present, but is capable of switching to fermentation or anaerobic respiration if oxygen is absent, and non-sporulating bacterium. Cells are typically rod-shaped, and are about 2.0 micrometers long and 0.25–1.0 micrometers in diameter, with a cell volume of 0.6–0.7 cubic micrometers. E. coli stains gram-negative because its cell wall is composed of a thin peptidoglycan layer and an outer membrane. During the staining process, E. coli picks up the color of the counterstain safranin and stains pink. The outer membrane surrounding the cell wall provides a barrier to certain antibiotics such that E. coli is not damaged by penicillin. Strains that possess flagella are motile. The flagella have a peritrichous arrangement. It also attaches and effaces to the microvilli of the intestines via an adhesion molecule known as intimin. Metabolism. E. coli can live on a wide variety of substrates and uses mixed acid fermentation in anaerobic conditions, producing lactate, succinate, ethanol, acetate, and carbon dioxide. Since many pathways in mixed acid fermentation produce hydrogen gas, these pathways require the levels of hydrogen to be low, as is the case when E. coli lives together with hydrogen-consuming organisms, such as methanogens or sulfate-reducing bacteria. In addition, E. coli's metabolism can be rewired to solely use CO2 as the source of carbon for biomass production. In other words, this obligate heterotroph's metabolism can be altered to display autotrophic capabilities by heterologously expressing carbon fixation genes as well as formate dehydrogenase and conducting laboratory evolution experiments. This may be done by using formate to reduce electron carriers and supply the ATP required in anabolic pathways inside of these synthetic autotrophs. E. coli have 3 native glycolytic pathways, EMPP, EDP, and OP. The EMPP employs 10 enzymatic steps to yield 2 pyruvates, 2 ATP, and 2 NADH per glucose molecule, while OP serves as an oxidation route for NADPH synthesis. Although the EDP is the more thermodynamically favorable of the 3 pathways, E. coli do not use the EDP for glucose metabolism, really. Common subdivision system of E. coli, but not based on evolutionary relatedness, is by serotype, which is based on major surface antigens, O antigen, part of lipopolysaccharide layer, H flagellin, K antigen, capsule, e.g. O157, H7. It is, however, common to cite only the serogroup, i.e. the O antigen. At present, about 190 serogroups are known. The common laboratory strain has a mutation that prevents the formation of an O antigen and is thus not typable. Genome plasticity and evolution. Like all life forms, new strains of E. coli evolve through the natural biological processes of mutation, gene duplication, and horizontal gene transfer. In particular, 18% of the genome of the laboratory strain MG1655 was horizontally acquired since the divergence from Salmonella. E. coli K12 and E. coli B strains are the most frequently used varieties for laboratory purposes. Some strains develop traits that can be harmful to a host animal. These virulent strains typically cause a bout of diarrhea that is often self-limiting in healthy adults but is frequently lethal to children in the developing world. More virulent strains, such as O157, H7, cause serious illness or death in the elderly, the very young, or the immunocompromised. The genera Escherichia and Salmonella diverged around 102 million years ago, credibility interval, 57 to 176 Maya, which coincides with the divergence of their hosts, the former being found in mammals and the latter in birds and reptiles. This was followed by a split of an Escherichia ancestor into 5 species, E. alberti, E. coli, E. fergusoni, E. hermani, and E. vulnerus. The last E. coli ancestor split between 20 and 30 million years ago. The long-term evolution experiments using E. coli, begun by Richard Lenski in 1988, have allowed direct observation of genome evolution over more than 65,000 generations in the laboratory. For instance, E. coli typically do not have the ability to grow aerobically with citrate as a carbon source, which is used as a diagnostic criterion with which to differentiate E. coli from other, closely related bacteria such as Salmonella. In this experiment, one population of E. coli unexpectedly evolved the ability to aerobically metabolize citrate, a major evolutionary shift with some hallmarks of microbial speciation. In the microbial world, a relationship of predation can be established similar to that observed in the animal world. Considered, it has been seen that E. coli is the prey of multiple generalist predators, such as Myxococcus xanthus. In this predator-prey relationship, a parallel evolution of both species is observed through genomic and phenotypic modifications. In the case of E. coli, the modifications are modified in 2 aspects involved in their virulence, such as mucoid production, excessive production of exoplasmic acid alginate, and the suppression of the OMPT gene, producing in future generations a better adaptation of one of the species that is counteracted by the evolution of the other, following a co-evolutionary model demonstrated by the Red Queen hypothesis. Neotype strain. E. coli is the type species of the genus Escherichia, and in turn Escherichia is the type genus of the family Enterobacteriaceae, where the family name does not stem from the genus Enterobacter plus I, sic plus Aceae, but from Enterobacterium plus Aceae, Enterobacterium being not a genus, but an alternative trivial name to enteric bacterium. The original strain described by Escherichia is believed to be lost, consequently a new type strain, Neotype, was chosen as a representative. The Neotype strain is U541T, also known under the deposit names DSM 30083, ATCC 11775, and NCTC 9001, which is pathogenic to chickens and has an O1, K1, H7 serotype. However, in most studies, either O157, H7, K12MG1655, or K12W3110 were used as a representative E. coli. The genome of the type strain has only lately been sequenced. Phylogeny of E. coli strains. Many strains belonging to this species. The first complete DNA sequence of an E. coli genome, laboratory strain K12 derivative MG1655, was published in 1997. It is a circular DNA molecule 4.6 million base pairs in length, containing 4,288 annotated protein coding genes, organized into 2,584 operands, 7 ribosomal RNA, rRNA, operands, and 86 transfer RNA, tRNA, genes. Despite having been the subject of intensive genetic analysis for about 40 years, many of these genes were previously unknown. The coding density was found to be very high, with a mean distance between genes of only 118 base pairs. The genome was observed to contain a significant number of transposable genetic elements, repeat elements, cryptic prophages, and bacteriophage remnants. More than 300 complete genomic sequences of Escherichia and Shigella species are known. The genome sequence of the type strain of E. coli was added to this collection before 2014. Comparison of these sequences shows a remarkable amount of diversity. Only about 20% of each genome represents sequences present in every one of the isolates, while around 80% of each genome can vary among isolates. Each individual genome contains between 4,000 and 5,500 genes, but the total number of different genes among all of the sequenced E. coli strains, the pangenome, exceeds 16,000. This very large variety of component genes has been interpreted to mean that 2/3 of the E. coli pangenome originated in other species and arrived through the process of horizontal gene transfer. Gene nomenclature. Genes in E. coli are usually named by four-letter acronyms that derive from their function, when known, and italicized. For instance, RECA is named after its role in homologous recombination plus the letter A. Functionally related genes are named REC, REC, RECD, etc. The proteins are named by uppercase acronyms, e.g. RECA, REC, etc. When the genome of E. coli was sequenced, all genes were numbered, more or less, in their order on the genome and abbreviated by B numbers, such as B2819 equals RECD. The B names were created after Fred Blattner, who led the genome sequence effort. Another numbering system was introduced with the sequence of another E. coli strain, W3110, which was sequenced in Japan and hence uses numbers starting by JW. Japanese W3110, e.g. JW2787, equals RECD. Hence, RECD equals B2819 equals JW2787. Note, however, that most databases have their own numbering system, e.g. the Ecogene database uses EG10826 for RECD. Finally, egg numbers are specifically used for alleles in the MG1655 strain of E. coli K12. Complete lists of genes and the synonyms can be obtained from databases such as Ecogene or Uniport. Proteomics. Proteome. Several studies have investigated the proteome of E. coli. By 2006, 1,627, 38%, of the 4,237 open reading frames, ORFs, had been identified experimentally. The 4,639,221 base pair sequence of Escherichia coli K12 is presented. Of 4,288 protein coding genes annotated, 38% have no attributed function. Comparison with 5 other sequenced microbes reveals ubiquitous as well as narrowly distributed gene families. Many families of similar genes within E. coli are also evident. The largest family of paralogous proteins contains 80 ABC transporters. The genome as a whole is strikingly organized with respect to the local direction of replication, guanines, oligonucleotides possibly related to replication and recombination, and most genes are SER-oriented. The genome also contains insertion sequence, elements, phage remnants, and many other patches of unusual composition indicating genome plasticity through horizontal transfer. Interactome. The interactome of E. coli has been studied by affinity purification and mass spectrometry, at per megasiemens, and by analyzing the binary interactions among its proteins. Protein complexes. A 2006 study purified 4,339 proteins from cultures of strain K12 and found interacting partners for 2,667 proteins, many of which had unknown functions at the time. A 2009 study found 599